really a great pleasure to be here to talk about our work and our current thoughts on targeted protein degradation. Our lab actually is a structural biology lab, and part of our research really focus on these CRL E3s. Early on, we were interested in the assembly of these E3 ligases and basically the general architecture of these complexes. And then later on, we become more and more interested in how these E3 ligases recognize substrates and how they're regulated, how could they achieve substrate specificity, and then how we could actually manipulate substrate specificity through chemical matters. And I want to first to show you one slide. This was a structure that I was involved in determining back as a postdoc fellow, and that was a collaboration between Nikola Pavlovich lab with actually Pagano's lab. And I think you have already heard of the P27 that was introduced by Avram. And this is the very first time we got a structural glimpse of the entire SCF complex built upon overlapping subcomplexes we were able to solve back then using crystallography. And of course, after we saw this complex, there are a lot of burning questions in the field. And I would say that our later study really focused on this portion of this complex. Here you have P27 represented by a short linear motif in its monophosphorylated form that's recognized by SCIP2, that's studied by Michele and together with Avram, and with the help of adapter protein called CKS1. And back then, and I think you can almost sense from the talk that's presented by the two of them, that there seems to be a wide belief that most of the substrates of these E3 ligases are modified by phosphorylation before they gain high affinity to the substrate. And this structure actually echoes yet a simultaneously determined structure by Pavlovich lab, where beta-TRCP, another F-box protein, recognized beta-catenin, again through this short linear motif that's double phosphorylated on two serine sites. And this is also actually extensively studied in the Pagano lab. So we actually established our lab with these all already published. And our early contribution to the field was to actually solve the structure of a protein called DDB1, which we validated or maybe demonstrated to act like the adapter of this CRL4 machinery. And we saw that in the literature, there's a viral protein called SV5V protein that actually has been reported to hijack this machinery to degrade otherwise very stable STATs proteins, which are the signal transducers in the interferon pathway in order to achieve antiviral effects. And we thought this viral protein most likely mimic the action of endogenous substrate receptors of this machinery that was missing back then. And we actually collaborated with Randy Moon's lab in our department. Using proteomic approach, we're able to pull down a whole bunch of proteins that have WD repeats embedded in their sequence. And we named them DCAFs together with Wade Harbour's group, who made the same finding. And of course, somewhere here, or maybe not even here, there's a DCAF15 or Umbra1. And then down here, where back then we had no idea what this protein was doing. This is the famous cereblon that becomes highly relevant later on. But this is basically a study, early study our lab did using structural biology approach to delineate the missing fourth or maybe fifth CRL complex in the field. But really, again, I was telling you that our main focus was on the substrate E3 recognition mechanism. And we realized that in the literature, even though many substrates are modified before they gain high affinity to the substrate, to the E3 ligase, there are examples where the substrate doesn't have to be phosphorylated or modified by post-translational modification. And here's an example of a rather recent study we did. We show that a very simple diglycine C-end motif at the extreme C-terminal site of a polypeptide as a product of abnormal early termination that can be recognized by a substrate receptor called CALHDC2 in the CRL2 machinery. So in this example, the C-terminal tail is exposed due to abnormal processing of or process of translation, and that leads to a protein quality control mechanism to eliminate the substrates. So then are all substrates actually recognized by E3 ligase through this kind of short linear motif in the primary sequence? The answer is not. Actually, again, in collaboration with Pagano's group and really inspired by studies that was actually done elegantly by Luca Buccino in the audience, we actually were become interested in this F-box protein, FBXL3, which plays a role in degrading cryptochrome, which are the central players in circadian clock. And here I hope you can appreciate that the substrate protein cryptochrome is a globular protein, and it doesn't have a short linear motif that's recognized by the F-box protein. Instead, the F-box protein actually, this leucine-rich repeat domain, wrap around the entire half of the one side of the globular domain. And at the same time, actually, it's a short linear motif from the F-box protein itself inserted into the deep central pocket of this substrate protein, which actually is known to bind to FAD. And at the same time, they actually occupy the same site where the obligated binding partner of cryptochrome called period is interacting with. And so this actually allowed both a metabolic cofactor and obligated binding partner to regulate this substrate E3 interaction. We later, in recent years, stumbled upon yet another F-box protein, FBXL5, where the substrate also is recognized through a globular surface instead of a short linear motif. In this case, FBXL5 is known to degrade a central player called IRP protein, iron responsive protein, in the iron homeostasis pathway. And in this case, you can see the IRP2 protein adopt a very large globular fold, and that's actually recognized at one side by the FBXL5 protein. And remarkably, in the literature, it's known that FBXL5 would only recognize IRP2 when there's enough iron or when the cell has enough oxygen. And it wasn't known what is the mechanism that conferred this iron and oxygen sensitivity to FBXL5's function. By serendipity, we actually discovered this FBXL5 actually sports an iron-sulfur cluster near the surface or near the interface between the substrate and E3 interface. And this iron-sulfur cluster is absolutely required for this FBXL5 to recognize its substrate. And it has to be in the oxidized form. That's why you need oxygen to sort of activate this E3 ligase. And in essence, actually, a single electron is dictating this interaction. And that's sort of a partially answer maybe the question Dr. Lin raised early on. What regulates E3 ligase? Here's an example. Oxygen and iron regulate E3 ligase by itself, okay? So these two examples sort of transformed our understanding of substrate doesn't have to be recognized through a short linear motif in the post-translation modification dependent manner, but you have actually more complex surface of a substrate that can help E3 ligase to achieve high specificity. But recently, actually, Michele, in collaboration with Michele's lab, we actually stumbled upon yet another fascinating system where now the substrate is recognized through its quaternary structure, not only tertiary structure. So this is unpublished results. And Michele's lab actually identified, published a paper early on FBXL22 as E3 ligase recognized or degrading BAH1. And we determined the structure of this complex and showing that BAH1 contains this N-terminal BTB domain that form a homodimer. And the diagram, so to speak, that's embedded in BAH1 is actually represented by this interface between the homodimeric BTB domain And this alpha helix beta strand constitutes the surface that's recognized by FBXL2. So now you have to have oligomer of substrate that can present the proper surface for F-box protein to recognize. And even more fascinating is we realize that BAH1, of course, is a transcription repressor in oxidative stress response pathway. And in the context of certain oxidative stress, actually this diagram has two conserved cysteine residues that can be modified by oxidative stress. And if these two cysteine residues are modified, it will perturb the local structure, thereby allowing BAH1 to evade the recognition of IPXL22. But then BAH1 is still degraded in response to oxidative stress, even though these two cysteines are modified. Why is that? It's because there's yet another F-box protein, FBXL17, that can kick in. FBXL17 doesn't recognize this diagram, but FBXL17 actually recognize or maybe interrogate the structure integrity of this BDB homodimeric assembly. In other words, actually, if this assembly is weakened, or if the protein-protein interaction is weakened, actually we found that two copies of FBXL17, together with the rest of the SCF machinery, would be engaged with the BDB domain of BAH1, and then remodel it, separate the two copies of BDB domain, and then form very stable monomeric complex in order to promote the degradation. So in this case, the diagram now is actually elevated to perhaps a fourth dimension. This fourth dimension is the structure integrity of a protein complex, but not necessarily the surface of a quaternary structure. Okay, so I hope actually just by this short introduction, I can show you using structural biology, we actually could understand how substrates are generally recognized by E3 ligases through the primary sequence in the form of short linear motif, either modified or not modified, or the tertiary structure fold that's presented by globular domains of the substrate. But now we actually found the landscape of a diagram is even more complex. It can be actually embedded in the quaternary structure of the substrate. And even further on, it can be the stability of the quaternary structure that are the signature motif that's recognized by FBXL, in this case, FBXL17. So I think along the way, we also found other cellular factors, oxidative stress, oxygen level, iron availability, all these endogenous or exogenous signals can actually come to play to regulate this interaction in a way that explains the complexity of biology, but at the same time, perhaps also present additional opportunities for us to come up with therapeutic compounds to manipulate these interactions. Of course, I want to highlight that all these studies, we are just the structural biologists, and it really relies on organic and intimate collaboration between us and multiple groups around the nation, if not the planet, to make sure that we are on the right path. But perhaps among all these studies, our most unique contribution to the field is the mechanism of action of auxin, which is a natural plant hormone that regulates every aspect of plant biology. And its discovery can be traced way back to Darwin, who actually suggested the existence of this small molecule in phototropism. Later on, it was identified as a very simple tryptophan derivative, chemically known as indo-3 acetic acid, that actually foster interaction between the F-box protein found in plants, already introduced by Michele, and in the presence of auxin, these transcription factors or transcription repressors called oxiaase can be recognized by tier 1 and degraded, thereby altering transcription program. And we actually used crystallography, again, many years ago, before cryo-EM revolution happened, to determine this very fascinating complex, showing that, by serendipity, we discovered inositol phosphate in the middle of this protein. But really, our structure reveals how the diagram, oxia is recognized by tier 1 protein in the presence of auxin. And auxin literally sit in between two proteins, fill up a gap to extend the protein-protein interaction and then enhance their binding. What we realized back then is that auxin is not a allosteric switch, which usually, let's say, a ligand binds to GPCR, alter conformation of the GPCR, allow GPCR to do something. It's not doing that because we solved the structure of ligand and ligand bound and ligand free form of tier 1 without auxin, and we don't see any conformational changes. Auxin is also not a PROTAC, a bifunctional molecule that was mentioned by Michele, and it was, the concept was developed by Craig Cruz and Rita Chase early on before this structure was reported. It's not a bifunctional molecule, but instead a very simple compound that's pretty much like a tryptophan amino acid sitting at the protein-protein interface. So we had to come up with a new term called molecular glue to explain auxin's mechanism of action. And of course, needless to say that sort of reflecting how fast science progress, in 3 years, Itos et al. At the Hirashi Honda's lab actually reported the target of thalidomide and or its derivatives collectively known as IMIDs. And then thanks to groundbreaking studies from Ben Eber's lab and Bill Kalin's lab from Dana-Farber Cancer Institute, they identified the target of thalidomide while it's reprogramming cereblon. So here we have the first example of molecular glue happening in human cells through therapeutic compounds. And of course, elegant studies from Nicole Thomas' group and from Phil Chamberlain's group at Celgene, they reported how cereblon works. They literally recapitulate the molecular glue mechanism as we showed for auxin, that they resurface the E3 ligase cereblon and then allow, together with the compound, the E3 ligase to recognize glycine loop from different neo-substrates. And these studies were later echoed by, I would say, almost a firework of discoveries of indisulam interacting with or reprogramming DCAT15 to degrade RBM39, which I believe James will talk about after me. And then other compounds like CDK12 inhibitor that would actually allow this cyclin K, CDK12 complex to bypass DCAF, directly glued to the DDB1 substrate receptor adapter proteins. And these really electrify the field and raises the question that can we actually prospectively discover target-centric molecular glue as protein degraders? This slide echoes what Michele already showed, and we're really fascinated by this burning question, how to come up with a rational approach to discover molecular glue degraders. Early on, actually, even before IMID story come out, we have thought about it quite intensively, and then we come up with this realization that perhaps we could, back then, this is without IMID's story, we could actually learn from auxin to do something like this, okay? Is that we know in human cells there are several E3 substrate pairs whose either mutation or defects in post-translational modification would lead to disease state where substrate can no longer bind to E3 ligases. Can we actually learn from auxin to come up with small molecules that can repair or maybe resurrect the interaction in order to overcome the effect of mutation that weaken the interaction, or maybe bypass the requirement of phosphorylation? Perhaps that's actually echoes what Michele just talked about, the cyclin D complex, cyclin D Ambra complex, to achieve enhanced interaction between substrate and E3 ligases to accelerate ubiquitination process. And I would say, actually, if you read the literature, Nurix did a fantastic job in this arena, targeting endogenous E3 substrate pairs, where they come up with a synthetic compound, Nurix compound, that can allow hypophosphorylated beta-catenin peptide with only one phosphate modifying the diagram to gain high enough, if not even higher affinity to the E3 ligase beta-TRCP, even though the monophosphorylated one itself has rather weak affinity. So I think this is really a proof of concept that we can, using MedChem, to push or perfectionize a small molecule so that it can achieve the effect of a covalently linked phosphate group or any other amino acid. So this is one obvious direction that I think we call a Tier 1, Tier 2 program, where we can exercise the molecular glue concept. But really what's more exciting and more promising is to think about how we can come up with chemical compounds that can foster new interactions between E3 ligases and, new interactions between E3 ligases and substrates that are not naturally degraded by the substrate, by the E3 ligase. So we thought about this after the EMIT story come out, and we really couldn't, I would say, figure out how to do this, because this is completely uncharted territory. And I would say early on, we had rather heroic, crazy ideas of exploring the chemical space and see whether we can come up with any compound that can foster interaction between any two proteins. Maybe back then we were very optimistic, and that certainly wasted a lot of time. We had to actually really come down to earth and think about something that's based on science, but not just enthusiasm. And one day, actually, I just happened to talk to one of my colleagues, Liang Cai Gu, who's in the biochemistry department, and he was trained in George Church's lab, and back then, he was very interested in come up with what's so-called dual nanobody chemical sensors. So basically, the concept is, yes, Baker's group can design, binders of small molecules, but would he be able to use basically random screening to look for nanobodies, two nanobodies that can only interact with each other in the presence of a small molecule? Then you can use these nanobodies as sensors for that small molecule. And in this case, actually, he was focusing on cannabinol, which is CBD, and his approach is rather straightforward or very conceivable. He would actually immobilize the ligand first, and then screen for nanobody libraries that contains many variants because they harbor random sequence in 3 loop regions, the complementarity determining region. And he would actually find, he was able to find actually nanobody variants that can bind to the ligand. He then immobilized this nanobody, the first binder, and then he would screen that library again, ask whether he can find a dimerization binder that would only interact with the first binder in the presence of the small molecule, right? So that's actually quite conceivable that he would be able to find something. And he did. He actually found many pairs of any small molecule. In this case, he can find a pair that the 2 nanobody would interact with each other in a CBD-dependent manner, and they wouldn't actually do it in the presence of a close analog, THC, okay? Despite the high similarity between the 2 compounds. So I was actually intrigued by his presentation, and then I was stunned by the fact that his drawing of this 2 nanobody system is so similar to the way me and Michele think about molecular glues. So I thought, wow, so in your case, you are probably really looking for the opposite, the answer of the opposite question is, if you have a glue, can you find the 2 proteins that the glue can foster interaction, right? So it must be a glue system. He said, no, this is not a glue system, because in his mind, even though he drew it this way, he thought it must be an allosteric mechanism. That is, the nanobody, upon binding to the ligand, would undergo conformational changes so that the second nanobody can bind. Even though he drew it in this simple way, he wasn't thinking it's a glue. So we kind of a debate with each other, back and forth, and eventually he said, well, Ning, if you think it's a glue, you better review the mechanism by determining the structure. I was like, okay, then we have to determine the structure. So we will use an experimental approach to determine the structure of the system. And lo and behold, it's a glue. So you can see that the crystal structure of this 2 nanobody together with CBD shows that the CBD is sandwiched in the middle, just like the way he drew it and just like the way I draw it. And And then the ligand is interacting with both nanobodies very extensively. But in order to really understand this system, we actually have to do some thermodynamic measurement, right? Try to understand the binding events of the system. So the advantage of this system, glue system, over other systems is that these nanobody proteins are very easy to purify. You can get bucket load of these proteins with the rotation student, but literally, right? You don't need poor postdocs to solve this problem. So we actually purified tons of these proteins, and then we did binary interaction among these three molecules for every pair. And consistent with the selection process that Liang Cai's lab used to find these nanobodies, the small molecule CBD has measurable affinity to the first ligand binder. Okay, this is the first binder he identified. And the affinity is 2 micromolar, so the affinity is not very, very high. And of course, he didn't select for a second dimerization partner for binding the small molecule. So when we measure the interaction, there's no affinity whatsoever. And then, of course, in the presence of the small molecule, we can find very tight binding between the 2 nanobodies. This is all consistent with what he hypothesized. What's inconsistent with his rationale is that because we were able to purify these proteins in large quantity, we actually were able to measure the affinity between these 2 nanobodies. Remember, in his screening process, he actually eliminated the second dimerization partner if it doesn't have, if it has affinity to the first binder, right? He has a counter screen after selecting the second nanobody that can bind to the first nanobody in the presence of the ligand. He would have a counter screen to eliminate those that can bind without the ligand. But turns out that his counter screen selection process, he didn't use high enough protein concentration. So he was measuring interaction at the 5 micromolar, and we were able to titrate it way up. So in fact, when we look at this interaction, the 2 nanobodies can interact with each other in the absence of the ligand. This actually is very highly consistent with the fact that the crystal structure show extensive protein-protein interface outside the ligand binding pocket. Okay. So this actually was very, very interesting because this was completely unexpected. If it's an allosteric switch, of course, they would not have any interface. But in this case, we saw interface, and we think this is a glue system, and that triggered us to think back to say, hey, wait a second, the known molecular glues, do they do the same thing? Are we missing something because we didn't do this kind of thorough studies? By all means, back then, in 2007, when we studied the auxin system, because the proteins are so difficult to purify, we didn't report any binding affinity whatsoever. We just report the structure. That was enough. But these days, of course, you have to report every parameter you can measure in order to publish in a leading journal. So this triggered us to went back to the drawing board and ask, is this phenomenon true for all known molecular glue systems? Lo and behold, when we obtained all these binding numbers for other systems, for example, in tier 1 system, auxin actually does have affinity to the E3 ligase, tier 1, although the affinity is 13 micromolar. It has no affinity to the diagram peptide, oxi-A peptide. That's expected because the peptide doesn't have any binding pocket. When auxin is present, of course, the peptide and the protein binds at 20 nanomolar. But what's interesting is that if you push the concentration of the peptide high enough, you can see endogenous basal interaction between the peptide and the E3 ligase, even in the absence of auxin. This, literally, all these numbers recapitulate what we found for the nanobody system. And then we ask, what about the cereblon system? In the literature, people already know the IMiD compound can bind to cereblon with high affinity. And that's a rather unusual property of molecular glue. And that actually revolutionized PROTAC because now all of a sudden you have a ligand that can bind to a E3 ligase, endogenous human E3 ligase. In their system, obviously, the glue doesn't interact with the neo-substrate because the glycine loop doesn't have a pocket. But we were able to measure the basal interaction between these neo-substrates and cereblon, even though these substrates are not the natural substrate of the E3 ligase. And what the small molecule does is to push these weak interactions into double-digit nanomolar range so that the interaction will become productive. The system I introduced you early on, studied by Nurex, where they found a small molecule that can allow beta-TRCP to gain high affinity to hypophosphorylated beta-TRCP. In that system, actually the small molecule doesn't have any affinity to neither the E3 nor the background substrate. And, but the reason they can do this is because the basal interaction is actually very strong between the hypophosphorylated peptide and the E3 ligase. And of course, their compound was able to do a really miraculous job by pushing the affinity more than 1,000-fold. That's actually higher than the effect where the second phosphorylation site can achieve. That gives us the hope that small molecules should be able to do the job just like these guys do to enhance basal interaction. So based on these studies, we thought we're in a unique position to kind of redefine molecular glue. We think molecular glues are very unique chemical inducers of proximity, different from PROTAC, by not showing affinity to at least one of the protein components of the system. And second is molecular glues action relies on the basal interaction in micromolar range between the substrate and E3 ligase. And the glue's job is to push this interaction into nanomolar range. In other words, you convert non-productive weak protein-protein interaction into productive strong interaction. And here I want to further highlight what do I mean non-productive micromolar weak interaction. So in my mind, these are what's so-called non-specific interactions. And if you say non-specific interactions, immediately it should ring a bell in your brain that, oh, there are sticky proteins that would bind to everything on this planet, right? That's one type of non-specific interaction. And in those type of non-specific interaction, your binding partners can actually associate with each other in all kinds of different forms. And your protein, probably because of its greasiness, can even stick to the surface of your test tube. That's the classic trashy non-specific interaction that we want to avoid at all costs. But the non-specific interaction that can be gluable, in my mind, they're actually post-specific non-specific interaction, meaning that when the two proteins are non-specifically interacting with each other, even though the specificity so even though it's non-specific, there's still a specificity in terms of how the two proteins interact with each other. It's just this type of interaction is very transient, very weak, that in the cell, because the concentration of these protein components are so low, the KD is irrelevant under physiological condition. Okay? And what we're going after are these type of interactions. And the key question is, of course, how to identify these interactions. Literally, for glue discovery, we think that the burning question is to identify this type of micromolar post-specific, non-specific interactions. And then we want to come up with small molecules that can fill up the gap, or maybe to resurface the E3 ligase or the substrate so that the interaction can be enhanced by roughly 1,000-fold. Most proteins in human cells are expressed at a nanomolar concentration. So when we achieve this type of enhancement, we will make this non-productive interaction into a productive form. The advantage of molecular glue, again, echoing what Michele already said, is that your molecular glue doesn't have to have affinity to at least one of the proteins. And if you look outside the cereblon image system, actually exemplified by the oxygen case, your molecular glue doesn't even have high affinity to the E3 ligase. Okay? The E3 ligase binds to molecular glue also at the micromolar range. In the presence of a substrate, it would actually sequester the glue in the middle and make the binding or the potency of the molecule much higher. Okay, so that actually, we think, provides an anchoring point for us, or maybe a starting point for us to think about how to prospectively discover molecular glue, is to first address the question, which E3 ligase you should choose to match a target that hopefully has therapeutic value that already been demonstrated. So in this direction, we have already mentioned by Michele and Avram, we have more than 600 E3 ligases. That provides the opportunity for us to choose the right E3 ligases. But out of this shelf, it's like you walk into Walmart, you have too many choices, which one you want. Well, you better use some filters to select, prioritize these E3 ligases. There are many factors, for example, tissue distribution, subcellular localization, whether we understand the E3s, or if we don't understand the E3s, we might have high risk. All these factors, of course, and the druggability, yeah, but we think a critical factor we should think about is the basal affinity. If we can select the E3 ligase that has the right basal affinity, then we'll have a much higher chance to find a glue that can enhance the interaction. And of course, Michele also mentioned that the advantage of glue is that it doesn't have to display high affinity to one of the binding partner at least. So that actually put it at a huge advantage of targeting non-ligandable or intrinsically disordered proteins, many of which are found in the neurodegenerative disease area. Okay, so obviously, we made a very small step toward the rational approach. We say basal affinity perhaps is the critical factor we should consider. The next burning question is, how do you find this? If I have the answer, probably we don't have to have this meeting to think about it. So I think I would want to invite the audience to think about what are the approaches we can use to actually discover this kind of basal affinity. And my gut feeling tells me maybe we can look into classic approaches, just like everyone is saying. Biochemistry is one way to do this, but we probably need expertise from other cutting-edge field that's outside the degradation field. For example, proteomics, for example, computational approaches. Okay, we have to think kind of out of the box to figure out what is the most productive way to achieve this. Once we're there, the rest is just conventional small molecule discovery. Okay, with that, I want to thank a long list of trainees and collaborators, and particularly biologists like Michele Pagano and Avram Hershko, who really set the stage for us to do structural biology, understand the basis of fundamental principles governing protein-protein interaction, so that we can come up with therapeutic approaches taking advantage of these fascinating biological systems. And of course, we really benefit tremendously from funding from NIH and HHMI, and of course, close interactions with SEED and Eli Lilly and many other companies who are interested in this space. Thank you for your attention.